Hello, everyone. It's my pleasure to welcome you all to the last lecture of our online series, Genomics in Immunohematology. So, in today's lecture, we will be discussing and getting to know of how we use next generation sequencing and genome sequencing data from a single individual scale to a population specific or a population scale blood profiling. So before moving on with the details of the topic and contents of this particular lecture, let us take a very quick recap of what all we have covered in all other previous lectures of this particular online course. So taking a very quick recap, we have covered the complete evolution of our understanding of how the human blood groups have formed and evolved over the years. And based on this evolution in our understanding, we have also got to see about the different types of techniques which have been used to identify the blood type of an individual, starting from the techniques which have been used from the past for over decades, which still remain as the gold standard techniques, towards the present techniques which focus on medium throughput molecular techniques and the advent of next generation sequencing. So we also got to discuss how the advent of next generation sequencing has positively impacted the field of immunohematology. Having said that, now we stand at a point where we have a clarity on the usage of NGS-based screening techniques and its advantage in the field of transfusion. So we could clearly state that with the help of NGS-based screening techniques, we will be able to get this genome sequencing data. It can either be a genome-wide sequencing or exome-wide sequencing, with the help of which we will be able to comment on the complete blood type of a person. By the word complete blood type, we mean that we would be able to get the extensive blood profile of an individual, which comprises the phenotypes of all the 44 blood group systems rather than just the clinically significant ones. So now coming to the details of the topic which we are going to discuss for this lecture today, we are going to talk about the differences and similarities of human blood groups based on their ethnic races. So there have been multiple hypotheses which talks about this particular concept and focuses about the truths and myths about this particular concept. So as we all know, human blood groups are very old genetic indicators which have evolved over the millions of years. So there have been multiple reports and studies which states the global distribution pattern of these blood group antigens and variants depends or varies depending upon different environmental factors, which includes disease, climate, altitude, humidity, geographical distribution, etc. So talking about that, there are three major hypotheses which focuses on this particular topic. It talks about the emergence and evolution of human blood groups. The very first mention goes to the primary races hypothesis, which talks about the distribution of blood group with respect to the geographical distribution. So it basically divides the global population into three major races, namely the European people who belong to the European subcontinent, those who belong to the Asian part, and those who belong to the South American geographical distribution. It says that the blood group A is found predominantly in Europe, whereas in Asia, the predominance of blood group B is seen. Whereas when we come back to South America, O blood group is found to be the predominant one. So with this, the primary races hypothesis states that with respect to the geographical distribution, the type of blood group which is predominantly seen in that particular area also differs. And talking about the other two hypotheses, which puts an emphasis on the emergence and the divergence of these blood groups, the second hypothesis says that initially, in the ancient times, the only blood group which was prevalent was the O blood group, which means that there was no expression of either the A antigen or the B antigen. And with due course of time, people started gaining the genetic mutations with which the two major blood groups, the two major antigens started emerging in the individuals, which led to the A blood group and the B blood group, which further and with further evolution, different types of subgroups started evolving in these two blood groups. This stands as the second hypothesis, whereas in contrary to this particular hypothesis, there's another hypothesis which says that the initial blood group could have been the AB blood group, which stands for the expression of both A and B antigens. And with due course of time, certain genetic mutation would have got accumulated or would have got dwindled away, which led to the differentiation into just the expression of A antigens or just the expression of B antigen, that led to the A and B blood groups separately. And again, with due course of time, they got diverged into multiple subgroups, and finally they got converged into the O blood group. So this is another hypothesis which talks about how the blood groups would have diverged and converged over the years. So there are multiple such hypotheses which have focused on this particular area of the blood group differing with respect to different ethnicities. What is the reason behind that, and a lot. This particular area is still need to be explored in a greater detail. Having said that, now if we talk about genomes and extrapolating the genome knowledge over a population scale, we could clearly see that certain blood types are very unique to specific racial and ethnic groups. Considering the example of the Duffy and the Lutheran and the MNS blood group, we could see that the Duffy negative blood types and the U negative blood types are highly seen or very much prevalent in the African and the African American population when we compare it with the other populations like the Caucasians or the Asians. So, with this, one thing becomes very much clear. So, it's very much important to understand the large scale genetic heterogeneity of a particular population so that it could help us in the development of population specific donor registries. We would get an idea of what is the blood group which is very much prevalent in that particular population, so that it would stand as a foundation for developing the population specific donor registries. So the overall idea is, based on the genomic data, we will be able to identify the presence or absence of blood group variants, which we are going to use and we are going to extrapolate it for the entire population. So let us start discussing about this particular concept in a much greater detail. So talking about that, a few more examples to follow. If we take this distribution of blood types in the United States by ethnicity, the statistics provided in the year of 2021, we could see this particular pattern. So if we consider the example of B positive blood group, we could see that this particular blood group, blood type is seen in a higher percentage in the Asian population when we compare it with other populations like the Caucasians, the African Americans, or the Latino Americans. Similarly, it stands the same with the A negative blood type, whereas it's just the opposite. This A negative blood type is seen at a very low percentage when we compare it with other populations. Very low percentage in the Asian population when we compare it with the other populations. So we could clearly see that in certain ethnicities, the prevalence of a particular blood type can be seen higher or lower when we compare it with other global populations. So this is just an example of a survey which was done in the United States. So this will hold good for all the global populations too. So now combining both the concepts together, we know that NGS, the advent of NGS has led to a large scale improvement in the number of sequencing which is being done on a global scale, and this in turn has led to the availability of a large amount of public data. So, having this in one hand, now when we ask the question, how are we going to explore the global blood profiles? We are in need of two prerequisite knowledge or the basic idea. One is we need large scale population sequencing data sets so that we study those data sets which are very much specific to certain ethnicities or populations. Then we also need computational pipeline of how we are going to use this data and how are we going to compare with other populations. So when we quickly take a look at all the publicly available population data sets for each population, like for example, Caucasians, Middle Easterners, there are a lot of publicly available sequencing data sets available nowadays. Now, like for example, the 1,000 genomes and the NOMAD dataset predominantly encompasses the samples which belong to the Caucasian population. Similarly, if we see about the Africans, we have the Gambian genome variation project data, which is available publicly, and we have the Qatar genomes and exomes, the Greater Middle Eastern variome data, which belongs to the Middle Eastern population. Similarly, for the East Asian, we have the genome sequencing data available for the Chinese and the Japanese population. We also have the Southeast Asian populations, which encompasses the Malaysians, the Singaporeans, and certain native populations like the Orangasli population. So now that we have population specific data sets that are clearly available, the next step which we need to know is we need to put forward a standardized computational pipeline using which we can analyze these population scale data sets, and we would also be able to compare if they are similar or different from other global populations. So, the very first proof of concept study, which explored the population scale blood profile of a particular population, was put forward in the year of 2019 by Scoman et al. So, this proof of concept study was performed on the Western Desert indigenous Australians. So, the size of the study dataset was about 72 samples. So the whole exome sequencing data was available for these 72 individuals who belong to the group of indigenous Australians. So whole exome sequencing was performed. All the blood group variants were identified, like we discussed in our previous sessions of how to identify the blood group variants from the sequencing data. Following the similar method, blood group variants were identified. Their associated blood phenotypes were also mapped back to. So we will have an idea of what is the percentage of each phenotype seen in that particular population. Like, for example, the Duffy A phenotype is seen in how many individuals out of the 72 individuals which are used in the study. So with that, we will be able to have a proportion in hand. Like, for example, X percentage of the population carry this particular phenotype in this particular indigenous Australian population. So by doing this exercise, we will have an idea on what is the proportion of a particular phenotype seen in that specific population. And once we have this number in hand, we can perform the same in other global populations, and we can compare the percentages if it is similar or is it drastically different between the two populations. So the the workflow for this particular exercise is going to be very much simple. We will have the sequencing data, which is the input data. This can belong to any global population. So the first proof of concept study was done on the Australian population. Similarly, this can be done on any of the publicly available data sets. What we do is going to replicate the same pipeline which we had discussed in our previous classes. So we are going to take the input data, we are going to compare it with our reference data set, which contains all the genetic variations and their associated blood phenotypes. And once we are done with the comparison, we will be collecting all those variants which are associated with the blood phenotype. And with that exercise, we will have the overall blood profile prediction for that entire population. So this is the exercise which is precisely done when we look or when we extrapolate this computational pipeline to a population. So a quick illustration to explain the concept which we discussed right now is to have a very quick look at this particular scatter plot. So this talks about the frequency of distribution of blood group variants, which are seen in India, and we have compared it with other global populations. These global populations have been taken from the 1000 Genomes Project. So this includes the African, American, European, and the East Asian and the South Asian population. So if we take a very quick look at the legend, which is given in the top right corner, so all those dots which are seen in blue color or signify the variants that are found double the time in a greater frequency in the Indian population compared to the population to which we are comparing it to. Similarly, all the green colored dots specify those variants which are found at double the time lower frequency in India. And all the red colored dots specify those blood group variants which are uniquely found only in the Indian population. And all those gray colored dots signify those blood group variants which are found in the similar range of frequency between both the populations, that is Indian population and the population to which we are comparing it to. So in this panel, we have compared it with the overall 1000 genomes data set. We have compared it with the Africans. We have compared it with the Americans. So the x-axis talks about the frequency of those variants in the Indian population, and the y-axis talks about the frequency in the population to which we are comparing it to. So in this panel, we could see that the variant frequencies are very diversely distributed. There's no kind of a very distinct similarity is not noticed with the Indian population when we compare it either with the African population or with the American population. And if we compare it with Europeans, East Asians, and the South Asians, we could clearly see that in the South Asian panel, there is a streamlined flow of these dots, which signifies that the frequency distribution of the blood group variants in India is very much similar to that of the South Asian population. In addition to this, we also see a handful of red color dots, which signifies that although the blood group variant frequencies are very similar between the Indian population and the other South Asian population, we also have a handful of unique blood group variants which are seen only in the Indian population. So, the take home message here is that blood group variant frequencies show vast variations between the Indian population when we compare it with other global populations. So, in spite, there are some similar variant frequencies which we observe between the Indian population and the South Asian population. In addition to that, we also observe some handful of unique blood group variants which are seen only in the Indian population that are not observed in the other South Asian populations yet. So, this is a very quick pictorial representation of how we work on population scale genome or exomic data and then compare it with other global populations to get an idea of how similar or different is our blood group profile, differing from the other global blood group profile, or how similar it is with other global population blood group profiles. So this is the complete, this is kind of the backbone of the concept which we were discussing in our today's lecture, of how we are going to use the advent of next generation sequencing and the advantages it has bestowed in the field of immunohematology, that is to use the publicly available population scale sequencing datasets to have an idea on the similarities and differences of population scale blood profiles. In our next lecture, we will have a very detailed hands-on and a demo session on how are we going to perform these exercises, starting from very simple examples to some complex examples. So I hope you stay tuned for the last lecture of this particular online course, where we are going to do some tasks and exercises. In our next